Hello learners, in this video I'll be talking about the heme metabolism. This is part 1 of heme metabolism where I discuss you the biosynthesis of heme. Biosynthesis of heme. The biochemistry behind the heme synthesis. Heme synthesis takes place in bone marrow where majority of heme is synthesized and liver. A small amount is also produced in skeletal muscle. In bone marrow, approximately 85% of total heme synthesis takes place. In liver, we have approximately 15% of heme synthesis takes place. So bone marrow is the predominant place where the heme biosynthesis takes place. Liver is having approximately 15% and less than 1% of heme synthesis takes place in skeletal muscle. The heme which is produced in skeletal muscle becomes part and parcel of myoglobin, and the one which is produced in liver is involved in making of cytochromes. And the majority of heme which is synthesized in bone marrow is actually involved in hemoglobin. The pathway is same everywhere. The heme metabolism is partly mitochondrial and partly cytosolic process. Partly mitochondrial and partly cytosolic process. There are total 8 steps of heme synthesis. Total 8 steps are involved in heme biosynthesis, whether it is bone marrow or liver or skeletal muscle, the pathway is all same everywhere. And out of these total 8 steps, 4 steps are in mitochondria and remaining 4 steps are in cytosol. And out of 4 steps which are there in the mitochondria, 3 are there in mitochondrial matrix and 1 is going to be there in intermembranous space. I'll be talking to you about that. So 1 step is going to be at intermembranous space and remaining 3 steps will be in mitochondrial matrix, and 4 steps, other 4 steps are going to be cytosolic. So let's start with heme biosynthesis in liver, and this is mitochondria where the process starts, and this is cytosol. Mitochondrial matrix I have drawn. The precursor molecule for making of heme is succinyl-CoA and glycine, which condense to form you alpha amino beta keto adipic acid, alpha amino beta keto adipic acid, or intermediate which is transiently formed during formation of ALA. So in fact, we are talking about synthesis of ALA, which is delta amino. ALA is delta amino levulinic acid. But the ALA is not instantly produced. We have one intermediate, that's alpha amino beta keto adipic acid, and this reaction is catalyzed by ALA synthase 1. So ALA synthase 1 is producing ALA in liver with the intermediate formation of alpha amino beta keto adipic acid. This ALA synthase 1 enzyme is actually the rate limiting enzyme in liver in mature RBC. This enzyme needs PLP. PLP is vitamin B6. So we need to have vitamin B6 for making of heme. Now once ALA is produced, let's see what happens next. The ALA enters the cytosol, and 2 ALA is required in the next step, and 2 water molecule is lost from them, and we tend to have the formation of PBG. PBG is porphobilinogen. Porphobilinogen, it's a cytosolic process. As you can see, in the cytosol we have dehydration of ALA as to produce you PBG, and of course you can even guess the name of enzyme, that's ALA dehydratase, or you can even call it as PBG synthase, right? PBG synthase. This enzyme is important enzyme in heme synthesis, and this requires zinc for action and also the sulfhydryl group of the enzyme for catalytic activity. So now we have formation of PBG. In the next step, we require 4 molecule of PBG which undergo deamination, and 4 amino group is removed. 4 amino group is removed from all these 4 PBGs, and they tend to link to each other in a head to tail configuration to form you linear tetrapyrrole, which is known as HMB. HMB is hydroxymethylbelane. Hydroxymethylbelane. As you can appreciate, it's a linear tetrapyrrole, and this process is all cytosolic, as you can see very clearly. The name of enzyme you can even guess. It's called PBG deaminase, or otherwise you can call it as HMB synthase. Previously, this enzyme was called as uroporphyrinogen 1 synthase, but now this name is obsolete and they are no more using this terminology. To avoid any confusion, they are not using this terminology anymore. So this is PBG deaminase or HMB synthase enzyme, which is actually in certain literatures it's written it's acting as a rate limiting enzyme in developing RBC. Now once HMB is formed, the next step is formation of UPG3, which is catalytic process, which is enzymatic process. We also have conversion of HMB to UPG1, which occurs spontaneously without the need of any enzyme, and this UPG1, after getting converted to CPG1, is getting excreted from the body, and no more, it is like no further is getting utilized in the system. So UPG1 conversion is actually the wastage of HMB, I must say. The major use of HMB is when it is converted to UPG3 by one enzyme that is called UPG3 synthase. UPG3 synthase enzyme. UPG3 synthase enzyme, or otherwise called UPG isomerase enzyme. UPG isomerase enzyme. See, again, this is a cytosolic process. Now, next step, what happens? There occurs decarboxylation of UPG3, and we tend to have formation of CPG3. CPG3 is, see, UPG3 full form, if I have not told you, it is uroporphyrinogen 3, and this is UPG1, it's a uroporphyrinogen 1, and this is going to be coproporphyrinogen 3. Coproporphyrinogen 3. See, 1 and 3, you must be wondering what is uroporphyrinogen 1, what is uroporphyrinogen 3. So for that you should know, like, these are tetrapyrrole rings and we have substituents groups on these 4 pyrrole rings. If the substituents groups are symmetrically distributed, it is called one series of compound. If the substituents groups are asymmetrically distributed on tetrapyrrole ring, it's called 3 series of compounds. And 3 series is also at times called as 9 series. So we have the 3 series or 9 series of compounds which are involved in the heme biosynthesis, right? Now what happens, this coproporphyrinogen 3 which is produced then try to enter the mitochondria back. And in the intermembranous space, we have an enzyme which oxidizes it, and this enzyme is known as CPG3 oxidase enzyme, which oxidizes it and produces protoporphyrinogen 9. You can call it as protoporphyrinogen 3, that's not wrong. There are books which mention protoporphyrinogen 3 also for this particular protoporphyrinogen. You can call it as 9. Now this protoporphyrinogen 9 is undergoing oxidation to produce you protoporphyrin 9 in mitochondrial matrix, and the name of enzyme is going to be protoporphyrinogen oxidase, PPG oxidase, protoporphyrinogen oxidase enzyme. Now what happens in the next step of heme synthesis? The iron is incorporated in presence of ferrochelatase enzyme. chelatase enzyme, because it's a chelating the iron or the ferrous in the center of protoporphyrin 9 ring as to make you heme. And you can always say this ferrochelatase enzyme as a heme synthase enzyme. It's also called as heme synthase. At times they write heme synthetase, though that's a misnomer. So heme synthase is responsible for making of heme. This is how the heme is produced. So there are total, if I just summarize the steps, there are total 8 steps. And total of out of 8 steps, 4 are mitochondrial, 4 are cytosolic. So let's see which 4 are mitochondrial. So allosynthetase 1, we count it as 1. Allosynthetase 1, then we have CpG oxidase, then we have protoporphyrinogen oxidase, then we have ferrochelatase. These are 4 enzymes which are mitochondrial. 1 is intermembranous out of these 4, as I have told you, and rest 3 are mitochondrial matrix enzyme. The remaining 4 enzyme, starting with aldehyde or PBG synthase, then PBGDA minase, otherwise HMB synthase 1, 2. Then we have UPG3 synthase 3. This is UPG decarboxylase. Sorry, I forgot to write this. This is UPG3 decarboxylase enzyme, which is going to decarboxylate it. This is also cytosolic. So there are 4 enzymes which are cytosolic enzyme. So this is a little typical pathway. There are multiple steps, multiple intermediates. And as I've told you, the rate limiting enzyme is allosynthetase 1, not 2. See, the exact same pathway is there in the bone marrow also, but we have allosynthetase 2 at the starting point. Right, and that gene of allosynthetase 2 is found at X chromosome, and this gene of allosynthetase 1 is found at chromosome number 3. You must remember this, and this is acting as a rate limiting enzyme. Allosynthetase 2 doesn't act as a rate limiting enzyme. And you must know this pathway because this is very, very important. And in the next part of the video, I talk about the regulation of heme biosynthesis. So in part 2, I'll be talking about the regulation of heme biosynthesis. So till then, thank you very much. Take care.